Hello. Today we're going to do a case study that has to do with action potential, and it's called Escape from Planet Soma. Hopefully that made you laugh because Soma means body. It's the cell body of a neuron. And there's lots more puns like that coming. After a valiant but doomed battle in the distant Purkinje galaxy, you are captured by the Glialians, the most evil beings in all of the cerebral hemisphere. They imprison you in their outpost on the desolate planet Soma, from which no one has ever been known to escape. Chief Oligodendrog eyes you with glee. I've heard of your daring deeds, and I must say your bravado impresses even me. However, bravado is nothing if your little earthling neurons can't produce some obvious intelligence to go along with it. I've yet to meet an earthling who possesses both. You shrug nonchalantly. There's always a first time. You warily eye his neuron incapacitator gun. Sure, if it's a fair test, and if I pass, you have to release me. Oligodendrog considers for a moment. Very well, let me explain the test. When prisoners try to escape, we use a variety of methods based on neurophysiology principles to discourage them from trying again. My assistant will select several methods at random, and you must predict the terrible effects produced when that method is used. Predict correctly, and you earn your release. Predict incorrectly, and you experience the effects firsthand. He smiles and clicks his clawed toes on the floor. Fervently hoping you remember something from those dreaded neurophysiology lectures in A&P, you agree. Excellent. Chief Oligodendrog grins. Here's the first method. He barks into a small radio, and a small glialian enters with an enormous syringe. Oligodendrog explains that it contains a mutant gene for a voltage-gated sodium channel in nociceptive neurons. Injection of the gene will produce channels that are non-functional, with disastrous consequences. He then hands you the card below. Kindly transcribe your answers so there is no dispute about what you said. Here are the 3 questions that I want you to answer. Number 1, how will the non-functional sodium channels affect the signaling capabilities of a neuron? Well, number 2, what type of information do nociceptive neurons carry? And number 3, why would having this mutant gene be so terrible? So here's that question again, and I just highlighted like the important parts. So there's a mutation in a voltage-gated sodium channel in nociceptive neurons. So we don't know what those are, but it's a mutation in a specific type of neuron, and it makes the channels non-functional. So they're asking us, how will non-functional sodium channels affect the signaling capabilities? So normally the voltage-gated sodium channels will open, and then sodium enters the neuron and the cell depolarizes. If they are non-functional, that means that the sodium can't enter, which means the neuron can't depolarize, which means it can't send a signal. What type of information do nociceptive neurons carry? You probably had to Google this to figure it out, but they carry information relating to pain. So we have a whole variety of different receptors, and like each receptor does a different job. Like some carry information about pressure, others carry information about temperature, others carry information about pain, and that's the one we're focusing on here. Why would having this mutant gene be so terrible? Like, doesn't it sound kind of nice, the idea that you would never experience pain? The downside of it is that pain is a protective mechanism. That means it keeps us from seriously injuring ourselves. Like if you have a broken bone and it's healing, it hurts to move the bone. That prevents us from moving the area too much and potentially damaging it. Or if I set my hand on a hot plate, if it hurts, I take it off. If it doesn't hurt, I'm just going to let it sit there until the whole thing burns up. I wanted to share this story with you. A complete lack of pain sensation is rare, but does exist due to this mutation in a subunit of voltage-gated sodium channels. People with this mutation have broken bones, suffered scalding burns, lost the distal part of their tongues, and sustained injuries to their lips that required plastic surgery. Children in particular have difficulty learning what actions could be potentially serious or life-threatening. One 14-year-old died after he jumped off the roof of a house. Yeah, so really makes you appreciate your voltage-gated sodium channels and the fact that they can transport pain. And by the way, this whole case study, even though it's like silly, it's based on real events. So every single thing we're going to learn about actually happens to our neurons. Next one. Oligodendron peruses your hastily scribbled answers. Not bad, Earthling, but that was only one technique out of many. The small alien enters again, this time with a flask of fluorescent orange fluid. This is one of my favorites. We've engineered a synthetic toxin that destroys the myelin covering your optic nerves and motor neurons. Here to have a sip of our special orange juice. It's really quite tasty. He hands you another card and swirls the oily fluid. Number 1. What effect will the destruction of myelin have on the signaling capability of a neuron? Number 2. Explain why this occurs. Number 3. Explain what will happen to you if you are forced to drink the alien orange juice. So this time, we're dealing with a toxin that's going to destroy the myelin sheath. So first question, what effect will the destruction of myelin have on the signaling capability of a neuron? So normally, myelin helps the signal to be able to travel faster. So if the myelin is destroyed, it will travel more slowly. And it might even travel so slowly that it actually dies out before it reaches the axon terminal. So in that case, the signal would not be sent to the next neuron. So myelin covers most of the axon, but it's not like one continuous band. It's like kind of like a bunch of individual bands together. I'm actually going to show the picture because me explaining this without the picture is going to be confusing. So this is our dendrites, and this is our axon, and this green thing right there is the myelin sheath. And then this is what I meant by how it doesn't cover the whole thing. Like there are these little gaps in between the myelin. Because remember, the myelin sheath is formed from individual cells, like the oligodendrites. And yeah, so they're not continuous. So that the nodes and the spaces between where the myelin sheath is, that is where we find like the voltage-gated sodium and potassium channels. So when we're trying to send a signal, nothing has to happen inside of the myelin. We only have to depolarize and repolarize here. And then the signal will jump to the next one, and it will depolarize and repolarize here. And then the signal will jump to the next one, and it will depolarize and repolarize here. If there is not myelin, you have to depolarize and repolarize along the entire length. So think about this, like depolarize, repolarize, depolarize, repolarize, depolarize, repolarize, blah, 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 long, long time, versus like depolarize, repolarize, depolarize, depolarize, because it's literally jumping, it's faster. Not sure if that makes any sense to you, but we'll see. Okay, and number 3, what will happen to you if you are forced to drink the alien orange juice? So we already know that the signal is going to travel slower, and we need to remember that he told us that it was going to specifically affect the myelin covering the optic nerves and motor neurons. Your optic nerves connects to your eyeballs, so this is going to be able to affect your vision. Like you wouldn't necessarily go blind, but you might lose some or all of your vision in one of your eyes or both of your eyes. And then motor neurons produce movement. So if it takes longer to send the signal to producing movement, that could mean that the muscles are going to be weaker, or they might even get to be paralyzed, depending on which way it affects. Number 3, Oligodendron narrows his eyes after reading this card and calls for his assistant again. Well, well, you've got a few neurons firing in that earthling head of yours, but we're not finished yet. His assistant enters, holding some sort of arrow with a sticky residue covering the tip. Sometimes we use a method borrowed from earthlings and prick uncooperative prisoners with an arrow covered in baracatoxin from a poisoned dart frog. This toxin causes voltage-gated sodium channels to open at a more negative membrane potential and also prevents their inactivation. An amount equivalent to a grain of salt will have nasty effects on your motor neurons. You smile as you take the card. This one should be a piece of cake. Number 1, how will the signaling of a neuron be affected if the voltage-gated sodium channels open at a more negative membrane potential? Number 2, how will preventing the inactivation of sodium channels affect the signaling capability of a neuron? Number 3, what nasty effect will this toxin have on motor pathways? So this time we're focusing on the baracatoxin, and it causes the voltage-gated sodium channels to open at a more negative membrane potential. So let's start there. How would the signaling of a neuron be affected if the voltage-gated sodium channels open at a more negative membrane potential? So in a normal situation, the threshold is about negative 55 millivolts, which means that the membrane potential has to go from negative 70 all the way up to negative 55, and then the sodium channels will open and then the membrane will depolarize. So if this baracatoxin is lowering the threshold, like let's say instead of negative 55, maybe it only has to reach negative 50 or negative 65, then it would be a lot easier to send a signal. So normally the resting membrane potential is negative 70, and it has to reach at least negative 55 or else it won't send a signal. But this toxin is lowering the threshold. So let's say it lowers the threshold to negative 65. That's really close to resting membrane potential. That means it's going to be super easy to send signals. We might even have neurons sending signals at resting membrane potential. The toxin also prevents the inactivation of the voltage-gated sodium channels. So if we think about this, normally the sodium channels are going to close, and then sodium stops entering, and then the potassium leaves, and we have the repolarization. But in this case, the sodium channels are going to stay open, which allows sodium to keep entering, so the membrane will become more and more positive. So it's basically like just stuck in a depolarized state. It's going to continue sending action potentials for some amount of time. And then basically we can't ever get back to the negative 70. The cycle is going to stop. We're not going to be able to repolarize and send new signals. So short answer, first it's going to send lots of signals, but then it will not be able to send any more signals. And how do we think that this is going to affect our motor pathways? If you're sending lots of signals, that's what can cause like a muscle spasm, like too many kind of random contractions. But prolonged stimulation, like sending lots of action potentials, is going to use up all the acetylcholine. So that means like eventually the next time the brain tries to send a signal saying like, hey, muscle contract, it's not going to have any acetylcholine to be able to contract. So basically your muscles will not be able to contract anymore. And then let's remember that the diaphragm and the heart are both muscles, and that's how baracatoxin normally kills people, by stopping their diaphragm, which controls your lungs and lets you breathe, or your heart, which pumps your blood. So, no good. And again, like, isn't it insane that just because the sodium channels are stuck open, it kills you? Because the sodium channels are open, it keeps sending action potentials, it runs out of membrane potential, it can't send a new signal, you're dead. If you see frogs that look like this, avoid them. These are some of the poison dart frogs that produce that toxin. Number 4, a low growl rises from deep within oligodendrog. You think you'll get them all correct? Don't be so smug. This time, the assistant brings in a cage containing an enormous black mamba snake. Oligodendrog rattles the cage, which makes the snake open its inky black mouth and hiss angrily. We've purified dendrotoxin K from the venom, and injecting it will block your voltage-gated potassium channels in no time. That will wipe the smile right off your face. Or maybe it won't. He laughs and presents yet another card. You read the questions and smile broadly at Oligodendrog. Number 1. What effect will the dendrotoxin have on the signaling capability of a neuron? Number 2. What will happen to you if your motor neurons are exposed to this toxin? All right, so this dendrotoxin is blocking the voltage-gated potassium channels. So first question, what effect will the dendrotoxin have on the signaling capability of a neuron? So normally the voltage-gated potassium channels are going to open and then potassium is going to leave. If we're blocking them, that means that the potassium can't leave. If the potassium can't leave, then the neuron is not able to repolarize, and the neuron is basically going to stay stuck in that depolarized state. And kind of like the last time, it's going to keep sending action potentials and releasing extra neurotransmitters even when we don't want it to. What will happen to you if your motor neurons are exposed to this toxin? Again, kind of like last time, we're going to see an increase in muscle contraction, and this can cause muscle spasms or tremors. And then here's a black mamba snake. The muscle spasms might not kill you, but there's a lot of other toxins in its venom that might kill you. So avoid this one as well. Once again, the instruction from your fabulous A&P teacher pays off. Oligodendrog roars in frustration and summons his assistant. He enters with a hose and mask attached to a silver canister. Here we go. How about a little puff of general anesthetic like sevoflurane? It will open more potassium channels in neurons of the reticular formation in the brainstem, and you won't know what hit you. Oh, but be careful, it's not those voltage-gated potassium channels that are affected. You gulp. Other potassium channels besides the voltage-gated? Number 1, to what other type of potassium channel is oligodendrog referring? Number 2, what effect will opening more of these channels have on the excitability of a neuron? Number 3, what will happen to you when sevoflurane reaches the reticular formation neurons that control sleep and consciousness? Great. So this time we have an anesthesia, like the thing that puts you to sleep when you're going to have surgery. And they're telling us that it's going to open more potassium channels in this specific part of the brain. To what other type of potassium channel is he referring? So if it's not the voltage-gated channels, he must be talking about the leak channels. You know those ones that are just always open and that allow potassium to just kind of diffuse out of the cell? What effect will opening more of these channels have on the excitability of a neuron? So normally potassium has a higher concentration inside of the cell. So it will naturally try to flow out of the cell. If we open more of these channels, then lots of potassium is going to flow out. So more potassium will leave. When potassium leaves, the inside of it becomes even more negative. It hyperpolarizes, and it makes it harder to be able to send a signal. We're moving it farther away from the threshold. What happens to you when sevoflurane reaches the reticular formation neurons that control sleep and consciousness? So in number 2, we decided that the result would be that the membrane is basically hyperpolarized, so it's not very likely to send a signal. And now we're saying that the neurons that control sleep and consciousness can't send signals. So normally your reticular formation is always sending little signals that like keep you awake and conscious. So if those neurons can't send their signals, you're going to fall asleep. Hence, general anesthesia. Number 6, beads of sweat dot your brow as you return the card. Oligodendrog notices, not so confident on this one, Earthling. However, his brow wrinkles as he reads your answer and crumples the card in disgust. You got lucky with that one. I'll trip you up yet. He turns to his assistant and roars, "What's next?" It's yet another syringe. Okay, tell me what happens when we flood your brain tissue with potassium until extracellular potassium levels are 10 times what they should be. This time, Oligodendrog flings the card angrily in your direction. A knot forms in your stomach. The questions are getting harder. Number 1, how will increasing extracellular potassium affect the signaling capability of a neuron? Number 2, what type of cell normally regulates levels of extracellular potassium in the central nervous system? Number 3, what terror will this method produce if injected into your brain tissue? So this time we're making the extracellular potassium levels high. Extracellular just means like outside of the neuron. So how will increasing extracellular potassium levels affect the signaling capability of the neuron? So normally, the concentration of potassium is higher inside the neuron, which causes it to flow out. But if we're increasing the potassium levels, that means that we're essentially reversing the concentration gradient. So we're making the concentration of potassium higher outside of the neuron, which is going to cause potassium to flow into the neuron. A positive thing flowing in would depolarize the neuron, which makes it easier to send a signal. What type of cell normally regulates the levels of extracellular potassium in the central nervous system? That would be our star-shaped astrocytes, which is in charge of basically mopping up the extra potassium that's outside of the cell so that this problem doesn't happen. What terror will this method produce if injected into your brain tissue? So we said that the higher potassium is going to make it easier to send a signal. So if this happened in your brain would basically become too active and it would start sending too many signals. And that is what causes a seizure, like excess brain activity causes seizures. This high concentration of potassium is called hyperkalemia, and it's actually how we kill prisoners like through lethal injection, like for capital punishment. Except instead of adding it into the brain, we insert the potassium into the heart. It makes the heart basically contract too much, and then it dies. So again, like this is insane that an extra amount of potassium kills you because it messes up the concentration gradient. The answers come to you at the last second, and instead of becoming angry, Oligodendrog appears almost resigned. You sense an opportune moment and venture an offer. Suppose we do one more method, any method of your choice. If I answer it incorrectly, I am your prisoner for the remainder of my days. But if I respond correctly, I earn my freedom and you suffer the treatment. Oligodendrog considers for a moment, then grins slyly. I'll accept your challenge. Prepare to make yourself comfortable here on Planet Soma. He leaves the room for a moment and returns with a small vial and a syringe. This is something entirely new that no one else in the hemisphere has ever heard of. We've been working on it for months, and it looks like you'll be the first Earthling to test it. Your heart drops to your stomach. You've remembered your A&P material pretty well so far, but something completely new? Perhaps you've overestimated. Oligodendron interrupts your thoughts. Some types of epilepsy are caused by a genetic mutation that produces a voltage-gated sodium channel with a faster recovery from inactivation. You could probably tell me that this would increase the excitability or firing rate of the neuron and lead to seizure activity in the brain. However, we've created a sodium channel with a different mutation. It alters the voltage sensitivity of the sodium channel so that it only opens at more positive membrane potentials. Amazingly, it also leads to seizures, but we're not sure how. Since you seem to have such a thorough grasp of neurophysiology, perhaps you will enlighten us. Number 1, Oligodendron hands you the final card. You both stare at the vial and wonder who will be the recipient of its contents. Number 2, how will the excitability of a neuron be affected by sodium channels that open at more positive membrane potentials? And number 3, how does that lead to seizure activity in the brain? So let's take a look at, first of all, like the part that's not the question. But he's saying epilepsy, which is a disease where you have seizures, is caused by a genetic mutation that produces a voltage-gated sodium channel with a faster recovery from inactivation. Basically, after the voltage-gated sodium channel is inactivated, that means that it's going to have to be able to stay closed. So if it recovers faster, that's going to be able to send more signals because there's less refractory time. Basically, you can get back to -70 for the resting membrane potential faster. But now on to the current problem. We have a voltage-gated sodium channel that opens at more positive membrane potentials. How will the excitability of a neuron be affected by sodium channels that open at more positive membrane potentials? So normally, the voltage-gated sodium channels open when they reach a threshold of around -55 millivolts, but this is saying they're not going to open until they reach more positive membrane potentials. So maybe it's going to be as positive as -30, or maybe it's even going to be as positive as 0. Either way, it's going to be harder to send a signal. Just think about this. Normally, the membrane potential is down here at -70, and all it has to do is reach -55, but this mutation is making it not open until it reaches 0 or whatever. It's a lot harder to make the inside of the cell that polar, or that depolarized, I should say. If they open at a more positive membrane potential, it will be harder to send a signal. I don't know why I wrote that twice. How does that lead to seizure activity in the brain? Yeah, that's a weird one because last time we said that seizures were caused by excessive brain activity, but this time we're saying it's harder to send a signal. So something I haven't mentioned is that we have a mechanism in us to prevent seizures from happening. Normally we have inhibitory neurons that will prevent signals from happening. So in this case, if a seizure is being caused in an instance where it's harder to be able to send a signal, I would guess that it's those inhibitory neurons that are not able to signal. So again, normally we have neurons that inhibit signals. We have an action potential, and the result of that is to turn off the signal. What would happen if we got rid of our inhibitory neurons? Things would signal all the time, and it would be crazy, and it would cause seizures. And this is one kind of hypothesis about what could be causing epilepsy.